Biochemistry, as the name implies, is the chemistry of biology. It is the study of the structure and physiology of living things on a chemical level. Plants, animals, and single-celled organisms all use the same basic chemical compounds to function. Biological molecules, proteins, carbohydrates, lipids, and nucleic acids interact to carry out activities necessary to living cells.
Among these important biological molecules are proteins. Proteins, which allow for the execution of essential processes such as the regulation of chemical reactions, transport of substances, structural contribution, and muscle contractions, are composed of amino acids. Amino acids are organic molecules consisting of both an amine group and an acidic carboxyl group. They generally consist of both of those components along with a hydrogen atom and an R variable group. So I'm going to go ahead and draw an amino acid right here. So firstly, on the left we have the amino group, which consists of 2 hydrogen atoms and a nitrogen atom. And to the right of that we have the central carbon atom, which basically acts as like a bonding site for all the other components of the amino acid. And below that we have a lone hydrogen atom. And on the right we have, probably not the best color, I'm going to change the color. On the right we have the carboxyl group, which consists of a carbon atom, 2 oxygen atoms, and a hydrogen atom. Now above the carbon atom is an R variable group. So this section here is basically what's going to determine what the amino acid is. So it's going to determine what its function and structure is, and that's what varies from amino acid to amino acid. Amino and carboxyl groups of separate amino acids link together via a condensation reaction to form a peptide bond. So basically this amino group of one amino acid and this carboxyl group of another amino acid are going to bond together and form a peptide bond, and this thus forms a chain of amino acids known as a peptide. And 2 amino acids together is a dipeptide, and so on is a polypeptide. So once you reach 50 amino acids linked together via peptide bonds, that's going to be considered a protein. So although proteins consist of a minimum of 50 amino acids, they can have as much as 1,000, and thus the possibilities of combinations are extremely numerous, with human cells making somewhere between 80,000 and 100,000 different proteins. Proteins take on a 3-dimensional shape determined by the interaction of folding of amino acids. Some proteins are globular or spherical, and some are fibrous or more linear. Protein shape can be disrupted in a process called denaturing, and if so, their function is changed or inhibited. Proteins serve as enzymes or biological catalysts, as means of transportation, such as in the case of hemoglobin, as structural support, and as hormones which allow for systematic cellular communication.
Carbohydrates are organic molecules that provide energy and structural material for living things. They consist of carbon, hydrogen, and oxygen in a ratio of 2:1 of hydrogen to carbon and oxygen. They can be found in foods such as pasta, fruit, milk, bread, and potatoes. There are 3 kinds of carbohydrates: monosaccharides, disaccharides, and polysaccharides. Monosaccharides are the simplest carbohydrates and are often called simple sugars. They consist of glucose, or what is commonly referred to as blood sugar, galactose, and fructose, or fruit sugar. Monosaccharides exist in both open chain and cyclic structures. The cyclic structures are more stable and are the predominant form of monosaccharides at equilibrium. Disaccharides are a combination of 2 monosaccharides produced by a condensation reaction. Sucrose, for example, is a disaccharide that is formed by glucose and fructose. Polysaccharides are complex carbohydrates that contain 12 or more monomers. There are 3 important polysaccharides: glycogen, starch, and cellulose. They all are composed of glucose subunits but have different properties and functions. Glycogen is found in animals and stores energy in areas such as the muscles. Starch and cellulose, however, are found in plants. Starch is used to store energy, while cellulose is structural and can be found in the rigid plant cell walls.
Lipids, the third category of these primary biological molecules, are large, nonpolar structures generally composed of fatty acids. They function to store energy, to create cell membranes, and to regulate cellular processes. Fatty acids are long-chain carboxyl acids containing 12 to 14 carbon atoms. They're essentially the building blocks of lipids, although lipids are not polymers like carbohydrates. So fatty acids can be classified into two main categories, that being saturated and unsaturated fatty acids. So here we can see a saturated fatty acid, and below that an unsaturated fatty acid. Basically the distinction between the two lies in the existence or the absence of double carbon bonds. So here we see a double carbon bond, whereas in the saturated fatty acid we do not see that. And you've probably heard of saturated fatty acids in relation to health concerns. They're certainly more harmful in terms of heart disease and cardiovascular health, and they can be found in foods such as red meats and eggs. Alright, so there are essentially four types of lipids, that being triglycerides, waxes, steroids, and phospholipids. I'm going to briefly go over each one, just to give you kind of like a general understanding of which each one is and what it's made of. So I'm going to go ahead and start with triglycerides. So triglycerides are basically long chains of amino acids bonded to a glycerol backbone, which this glycerol backbone is the same as right here, and these amino acid chains are connected through ester bonds. That's just the name they're given. In the case of triglycerides, are just called ester bonds. So triglycerides are essentially the fats found in the human body. They're used for energy. It's just fat cells that are reserved for energy when other sources like carbohydrates are depleted. So next we have phospholipids, which basically makes up the most cellular membranes. So it allows for the regulation of what comes in and out of cells, because if they weren't there, then anything could go in and out of cells. So I'm gonna go ahead and draw a bilayer. So we've got the nonpolar heads of the phospholipids. So they consist of phosphate and lipids, as the name kind of implies. And these blue rings, obviously not the best rendition of a phospholipid bilayer, but these blue rings are the nonpolar heads, and so this is the outside of the membrane. And on this top side you'd have like extracellular fluid. Below that you'd have the inside of the cell, which is the cytoplasm, and you have the nonpolar tails right here, and the polar tails right here, and the nonpolar heads right here. So that just creates a phospholipid bilayer, which, like I said before, is the cellular membrane. So I'm going to move on to waxes. Waxes, it's not really much to say. They're just, for example, in candles or on the coating of leaves, they can prevent water loss. Alright, so steroids are generally going to follow this ring structure. They're pretty important in the human body and in most organisms. They consist of, hormones, vitamins like vitamin D. So they basically allow for, like, hormones, for example, allow for communication and, let's see, let me try and focus on this. Alright, so they allow for communication and the regulation of like certain processes, like metabolic processes and whatnot. So this is a steroid, and yeah, those are the four types of lipids. So we've got triglycerides, phospholipids, waxes, and steroids, all serving unique purposes. And we're going to move on to the last biological molecule, which is nucleic acids.
Nucleic acid is an organic molecule that composes DNA and RNA and is involved in the storage and transmission of genetic information. Nucleotides make up nucleic acids. Each nucleotide has an inorganic phosphate group, a 5-carbon monosaccharide sugar, and a nitrogenous base. DNA is one of the 2 types of nucleic acids found in living cells. The structure of DNA is a double helix that resembles a twisted zipper. It contains 2 long chains of nucleotides. Each nucleotide contains a phosphate group, the 5-carbon sugar deoxyribose, and a nitrogenous base. There are 4 different nitrogenous bases: adenine, thymine, cytosine, and guanine. Adenine and thymine always pair, forming 2 hydrogen bonds between them, and guanine and cytosine always pair, forming 3 hydrogen bonds. The pairs are called complementary base pairs. DNA stores the genetic information of a cell in the cell's nucleus. The sequence of the 4 nitrogenous bases of DNA give instructions to the organism. RNA and DNA differ in their components. RNA contains adenine, cytosine, guanine, and uracil. Thymine is never found in RNA. All DNA contains the sugar deoxyribose. RNA contains the sugar ribose. The structure of RNA differs from that of DNA since RNA is usually single-stranded, with no such hydrogen bonds forming among the bases. Whereas DNA functions to store genetic information, RNA allows cells to use the information found in DNA.
Plants.